It's been a great meeting, and I was very happy to come back and see some old trainees, Margaret Ricard, Roxanne Tusiwan, Barbara Schramm, are all in the audience, so it's great to see you here. Here's the current lab, and today I'm going to speak just about 3 short little vignettes, one by Fayetto, some things by Canton Liu, and something by Luke Costo in the lab. So, this is the title I usually give, Dendritic Cell Development and Function, but I think it was Charlene who first introduced the concept that we've heard about all for the whole meeting, and that's the concept of how complicated and how we chronically underestimate the difficulty and complexity of the problems that we're trying to solve. So I rename my talk and reword it as the things we don't understand about dendritic cell development and function. Now, being last, I know we're all exhausted, and I am, and so I need your help today to give my talk. So it's going to be audience participation, and I will let you know when you're supposed to say your lines. You see my lines? What are my lines? Well, I don't know if everyone's seen this movie, but it's a great movie by the Coen brothers, and I'm particularly interested in this one scene between Walter Enright and Ralph Fiennes, and it's a scene you see that Ralph Enright is playing a young actor in a movie, and it's a movie about a movie, and his girlfriend is cheating on him, and he comes to the party, and he's just seen his girlfriend and the other guy leave in a car together, and so he comes into the room, and Deirdre says, "It seems like there's a no-show, which is simply a bore, but I can partner with you at bridge." "Oh, why the pout?" And Hobie says, his line, "Would that it were so simple." And at that point, Ralph Fiennes, whose name in the movie is Lawrence Lawrence, says, "Cut." And he brings Hobie up to him and said he's going to try to teach him how to say, "Would that it were so simple." And here's the
Right, let's try it. Your line, just say as I say. Say your line exactly as like that. Just like that. Sure, I'll go. With the decursives. Would that it was so simple. With the decursives. Would that it was so simple. I think I used it to what did you say like I said? Would that it was so simple. Would that it was so simple. Would that it was so simple.
All right. Now, those of you who aren't native speakers of English probably are confused by the would that. This is the archaic Middle English form of the subjunctive mood. And so in modern English it would be, "I wish it was so simple." Or if you're a little younger, you stay down till 3 o'clock and you're a little skeptical, you just say, "Yeah, right." So, now, audience participation, let's practice for a minute. I can't hear you. One more time. This time, just the boys. Ladies.
We're so simple.
That's much better. Okay. So you'll see, I'll give you your cue when it's necessary. So it's about BHF3. So BHF3 is a strange AP1 factor that forms a Jun dimer and then specifically interacts with IRF4 or IRF8. And in dendritic cells, it interacts with IRF8 and binds to this particular particular sequence called an AICE. That is a defined element that's specific for this heterotrimer complex. A BHF knockout mouse lacks dendritic cell development because the terminal DC1 can't maintain IRF8, because the IRF8 gene is controlled by this plus 32 kb enhancer that we identified. And knocking out that enhancer using CRISPR, just a short bit of it, gets rid of all DC1s in that box there. And it's almost as good as IRF8 knockouts, and it's much better than BHF3 knockouts because you're not disturbing the expression of BHF3 itself, which is sometimes expressed in other types of cells. And then finally, this 32 kb enhancer is the region where IRF8 expression interacts with the BHF3 gene to maintain IRF8 expression, and that's how autoactivation maintains a stably on state in the DC1. But years ago, when we first identified this enhancer, these little green boxes, those are the predicted AICEs in this enhancer. And what we noticed really early in this work was that they're lousy. We identified it by chip seed, yes, but when we carried out what's the old-fashioned MSIP analysis, these sites don't bind. They can't even form a band. So the band under the CTLA4, that's a probe from the CTLA4 gene that's highly activated by this complex. The sorting exon 22 is a highly DC1 specific gene, and the enhancer element in that gene forms a really proud complex that you can see there. But none of the sites in the 32 kb enhancer are any good at all. And this is just an experiment where you use those sites in competition, and you can see by the band that goes across in that red box, none of the sites in the 32 kb enhancer compete effectively for formation of this IMSA complex there. So really the question is, why are the AICEs in the 32 kb enhancer so lousy? One of the ideas that we had was that maybe it's on purpose, because in the DC2 progenitor, I'm showing you here the CDP and pre-DC2 and immature DC2 in the periphery. There's a point where BHF3 is gradually induced during DC2 development, and IRF8 gradually extinguishes, but there is a point where they tend to cross over. And so we had this idea that maybe we could take advantage of this low affinity and make a mouse that didn't have any DC2s. So the hypothesis was that if we were to put high affinity elements into the 32 kb enhancer, then it would auto-activate in all the DCs, and you'd get a mouse that only had DC1s and never had DC2s. So Theta O made this mouse. He collected all the highest affinity elements that we could find, and we stuck them all together and used CRISPR to insert a large number of high affinity elements right into the middle of the 32 kb enhancer. And so we had no model of DC2 deficiency. We hypothesized that high affinity enhancers would make everything become a DC1.
We're so simple.
Thank you. So, now this just came out last week in Nature Immunology, so I'm only going to briefly show the data. It's not so simple. Instead of generating all DC1s, what happened was we generated yet a third weird population of DCs. We don't know what to call them, but we called them in the study hybrid DCs because even at the single cell level, this is just bulk RNA-seq, but the single cell level shows the same thing. These hybrid DCs that are, in this case, expressing XCR1 and SERP-alpha, these DCs express all the genes that are DC1 specific, but also all the genes that are DC2 specific. So what I'm going to show you in just a moment is this is the normal way that DCs develop. You have the pre-DC1, and it only develops into a DC1. You have the pre-DC2, which you can find in the bone marrow, and it develops into a DC2. But in this mouse, everything is scrambled. What I'm going to show you is that particularly the pre-DC2 specifies and then turns back into either a hybrid DC or a DC1 because of inadvertent autoactivation. So here's the data. You can see what I'm doing here is showing you that even if you take away DC1 specification, so this little box here is the pre-DC1, it's present even with one allele, but in the NFL3 knockout, it's gone. But and if you cross it to the high allele, this special enhancer, it's still gone. So there's still no DC1 specification. But what happens in the periphery is that the DC2 progenitors have turned into a hybrid DC, and some of them have turned all the way back into a DC1. So these are coming from the DC2. If you just purify the DC2 progenitor and put it in culture, the normal DC2 progenitor stays down in the SERP alpha DC2 gate. But if you have the high allele of high affinity 32, half of those or more turn back into a DC1. And the way to explain this is as follows. So this is the normal situation where a pre-DC1 has induced BHF3. The level of IRF8 and the high level of BHF3 combine to these weak low affinity enhancers. So that switches on and keeps the IRF8 gene expressed because the enhancers are occupied by complex. In a pre-DC2, there's very little BHF3 at that moment, and so there's not enough compound heterozygosity. There's not enough factor to bind these and the enhancer stays off. Whereas if we put in high affinity sites, even a little bit of BHF3 is apparently sufficient to occupy the sites and turn IRF8 back on when it's not supposed to. Now, a completely paradoxical and unexplained phenomenon that we also observe is that even in the DC1s, in these high mice, the IRF8 is not as high. We expected it to even be higher than the normal DC1, but in fact, it's lower. And if you look at the DC2s, they express higher levels of IRF8 than a DC2 is supposed to. So what does this do? Well, the answer is it completely ruins the ability of the DCs to support the modular immunity that we know they do. DC1s are supposed to support tumor rejection, and DC2s are supposed to support Th2 responses and maybe Th17 responses. So we did the H-polyvirus in collaboration with Mike Diamond in our place, and the granulomas that we're supposed to produce abundantly over here in a wild type are poorly produced in this high mouse, even though it has DC2s. And importantly, the tumor rejection is completely messed up. Here's a wild type rejecting some of these Bob Schreiber tumors, but in the high mice, they're just as bad as DC1 deficient mice. So, and the tetramer expansion is also deficient. So, the point is that we don't understand this. We asked the question, why were the affinities so low? It told us that they have to be low in order to cleanly segregate DC1 to DC2. If you try to improve on nature, you just make it worse. And then finally, this completely paradoxical finding that you try to make the enhancer better, and the gene expression gets worse. So this is probably going to be delving into some weird, highly technical transcriptional machinery loop model of enhancer function that we need to understand. Now, I was going to tell you another story about the F1 effect, but I don't think there was time. So instead, I'm going to go tell you something brand new. Florent, I didn't understand most of what he was saying, but I recognize the term ID2. So these are the factors that are required for DC1 development. And IRF8 was the first one, 25 years ago almost. ID2 was the second one. And then this other list of factors that all contribute to DC1 development in various ways. But ID2 is the factor we don't understand. So that's what I want to tell you today. It's not complete, and we think we have an explanation, but we'll see what you think. So this is showing the IRF8 enhancer locus. So this is the IRF8 gene. It's expressed that we've identified 3 distinct enhancers, the 56, the 41, and the 32. Now the 56, there's an inaccurate, there's a report in the literature claiming that the 56 enhancer is regulated by RUNCs. That's just incorrect. Our work, it's not published yet, but our work shows that this is actually, we've identified exactly 2 CEBP alpha binding sites. When CEBP alpha is expressed, it turns on the 56 kb enhancer, and this leads to the formation of MDP, because that then leads to MCSF receptor expression along with FLT3 that's already expressed, and you have an MDP. When the MDP transfers to the CDP, another set of factors take over at the 41 kb enhancer, and that's published, so I'll just remind you that those were E-boxes binding either E2A or E22. And then finally, when BHF is expressed in the specified 3DC1, that's when IRF8 is transferred over to the 32 kb enhancer. So what about ID2? So what I'm showing you here is what we know about DC1 specification is that NFI3 is induced and acts to repress ZEP2, in which BHF3 is expressed, and then you can become a DC1. But why is ID2 required? So this began a couple of years ago when we published this paper. This was the most complicated paper we've ever done. It involved epistatic analysis between these 3 genes. We had to look across conditional knockouts to other knockouts, and I can't go through the data, but I will show you what the circuit was that the interpretation of the epistasis made for us. So it placed NFI3 as the top factor on starting the process of DC1 development. Now, NFI3 is a repressor, and we concluded that it repressed ZEP2 somehow, either directly or indirectly, and that ZEP2 and ID2 had a weird relationship. They were both upstream and downstream of each other, so ZEP2 repressed ID2, but the data also suggests that ID2 epistatically suppressed ZEP2. So we interpreted this to mean that it might have an E protein supporting ZEP2 that was repressed by ID2. And all of that was just interpretation. So the questions we had to answer were, how does NFI3 repress ZEP2? And why is ID2 required for DC1 development? Now, when you think of myeloid cells, you typically don't think of E proteins. They're supposed to be myeloid. Now, here's an example. Here's a IL-7 positive lymphoid progenitor, and this is intracellular facts for E47, a form of the E2A gene. So, and on the vertical axis is CEBP alpha, and you can see that lymphoid progenitors express E2A, but not CEBP alpha. But if you look at the MDP by the same assay, MDPs are as bright for E47 as are lymphoid progenitors. And they now express high levels of CEBP alpha, as do the CEBP. So we should need to think about E proteins in the myeloid pathway. So at about this time, Xiao Huang in the lab was looking for the hypothesis of how the E proteins regulate ZEP2. And he found that by ChIP-seq of E proteins, a peak at minus 165 kb upstream of the ZEP2 gene. And we call this the minus 165 enhancer. And this was published a couple years ago. What he did was to knock out the entire enhancer, all 400 nucleotides, and this gave rise to the observation that PDCs and B cells didn't develop, which we could understand, but also that monocytes don't develop, which we didn't understand. And he also focused our attention just on the 2, 3, and 4 sites, because this last site, E1 over there, didn't bind E proteins at all. So we will only consider E2, 3, and 4 from here on out. A year or two later, Tian Lu in the lab was looking for how does NFI3 suppress ZEP2, and using ChIP-seq, and this is published, I'll just review briefly. She looked for ChIP-seq using NFI3, and she found a peak exactly in the same location as Xiao had found, except her sites for NFI3 were these sites that I'm showing in the red boxes. And those were clear NFI3 binding sites. But the surprise of this paper was that she also discovered that these same exact red boxes bind CEBP alpha and CEBP beta. Then we realized that these factors, the CEBPs, and NFI3 have almost exactly the same DNA binding motif, which was buried in the literature, and we haven't encountered that fact yet. And she also showed by just the old-fashioned MSA analysis that all of these red boxes directly compete for both binding to NFI3 and CDP alpha and beta. So she made this mouse where she selectively mutated the base pairs, not deleted, but mutated the base pairs for the 3 individual CDP binding sites. And we call this the 1, 2, 3 mouse. So the terminology, I'll try to be clear, triple mutant. We first called it the triple mutant, and the result of this is that we got the ablation of DC2s and monocytes, but we had normal PDC development and B cell development. Apparently the E protein binding sites are still present and completely support B cells and PDCs from the myeloid lineages. And here's the data for that. That triple mutant has no DC, almost no DC2s in the periphery, and the DC2 progenitor over here also fails to specify. So, and the reason for that is that all of the myeloid progenitors that would like to become a monocyte or a DC2 instead convert over to a DC1. So here's how we understand that. CDP is expressed and supporting ZEB2. When NFL3 is induced, it suppresses ZEB2 and BHF3 and ID2 come up, and that leads to DC1 development. But the puzzle that I'm going to address is Tian in the same paper had made an NFL3 GFP fusion protein, because you need to know whether the protein's expressed. And what she noticed down here is that CDPs have a fraction that are positive and go off and become a DC1, but by the time you even get to the identifiable DC1 progenitor in the marrow, NFL3 is gone. And so we interpreted this like so, that there's a pulse of NFL3 expression, and somehow that's translated into successful DC1 specification and commitment. So the real question is, why does the transient expression of NFL3 lead to, why doesn't it lead to just a transient suppression of ZEB2? So, hypothesis. Don't talk yet. ID was required to successfully repress ZEB2. And we would see that as the following. So, if ID2 were expressed, ID2, we know, forms a heterodimer with E proteins, but lacking a DNA binding domain, that complex cannot bind to the ZEB2 enhancer, which would lead to the loss of ZEB2, which would allow DC1 development. And so maybe the hypothesis is that in the ID2 knockout not being induced, E proteins still capable of binding to the ZEB2 enhancer, leading to the continued expression of ZEB2, preventing DC1 development. So, Fayette in the lab wanted to do this experiment. He wanted to selectively mutate the E boxes in the ZEB2 enhancer, and he predicted, he asked, if I cross that triple mutant of E sites with the ID2 knockout, would that now disallow ZEB2 expression or suppress ZEB2 expression and allow DC1 development? So to do that, he did the same thing with some help from Tian Lu, starting with the mouse that Tian had made, and knocked out all the 3 sites, and I'll call that the triple E mutation. So here's the triple E. It has the C boxes binding NFL3 and the CDPs, but it's lacking the 3 functional E boxes in the enhancer. And here's what we got. Some of it's predictable. We lack B cells and PDCs, and that's shown here, because the E protein support for ZEB2 is gone. But interestingly, it has monocytes. That's shown over here. So we lack PDCs and B cells, but we have monocytes. So now, let me summarize the 2 lines. Here's the original triple C mutant, and they have B cells and they have PDCs, and they don't have monocytes. And And the new mouse that lacks the E boxes has no B cells and no PDCs, and it has monocytes. So, these seem to be the opposite. So, the first mouse had no DC2s. So, they seem to be opposite, and the triple E box mutant should lack DC1, right? No, that's not what we got. It's not so simple. Instead, they act, for DC development, these two mutants basically act the same. So, you have the 123 mouse and the 234 mouse. Both have a substantial decrease in the DC2 specified progenitor in the bone marrow. Now, this is gated on the myeloid progenitor that you can find coming out of the CDP. Now, this is gated on the newly identified progenitors for the lymphoid branch of DC2, which we call the transitional DC-PDC-like DC2 progenitor, also published by Florent and others. But it's completely gone. So the DC2 specification from either branch completely relies on the E boxes in the ZEB2 enhancer, as you can see here. So the other thing that this mouse has is it has a substantial increase in the numbers of DC1 progenitors. This is our box for the DC1 progenitor in the bone marrow, and both of these have much higher numbers of that than the wild type mouse. But, okay, what about this hypothesis about ID2? So on the left, I'm showing you a wild type, an ID2 heterozygous mouse, and an ID2 knockout mouse. And what you can see is that the wild type can have DC1s, but you cross it all the way into the ID2 knockout, and the DC1s are gone. But now, you cross the triple E mutants onto all of those backgrounds. The wild type triple E has lots of DC1s. ID2 knockouts, they still have lots of DC1s. And now you can see that you cross the triple E to the full ID2 knockout, and the DC1s come back. And on the top is just spleen, and on the bottom is bone marrow cultures for DCs. Now the point is, so right there. So now I converge those so you can compare. Here's a wild type, just a normal wild type mouse. And there you have your typical fewer DC1s than DC2s in that box. But now we have crossed we've taken away ID2, but we've also taken away the 3 E boxes that ID2 targets in the CDP, and you get almost normal numbers of DC1s coming out. And when you culture in vitro, you almost get only DC1s. So we think that maybe ID2, I'm going to propose a model, and it may not be the final model, and it doesn't animate exactly the way I want it to. But, so we think in the myeloid lineage, you do have E protein support contributing to the ZEB2 enhancer, along with CDP, at different sites that are interdigitated, like the card game. And that's supporting ZEB2, and that's suppressing DHF3, so you can't become a DC1. And then, when NFL3 is transiently expressed, we think this leads to an active suppression at the enhancer, leading to the loss of ZEB2. And the next step after the loss of ZEB2 would be BHF3. But what if BHF3 was the only one induced and ID2 wasn't induced, like in the ID2 knockout? Then what would happen is when NFL3 goes away, you have E2A there. And that could reignite the enhancer and make it a transient suppression. And CBPs are also expressed, so they could chime in, and you get re-expression of ZEB2 and no DC2 at all. So if you have NFL3 suppression, and if that allows ID2 induction, ID2 can then go and ruin E protein DNA binding by heterodimerizing and blocking re-ignition of the enhancer. And so ZEB2 stays off. So that's what we think ID2 does in DC1 development. We still don't understand many things about this. How does ZEP2 repress ID2 and DHF3? We have tried and failed multiple times to identify ZEP2 binding sites in these genes, and we've targeted some. It's extremely disappointing to the enthusiastic students who try and fail. They graduate. We also think there's a complex biology to the repression, and Berger Singh has talked about that. And we have the enhancer for the induction of NFL-3, but we don't know what induces it yet. We have some suspicions. And the biggest mystery for me in this whole field is that NFL-3 deficiency is overcome by administration of FLT3 ligand. And we don't know how that works. And I have suspicions, but we really don't know. But I think that's a very important thing to consider therapeutically, because if there's gated DC1 development, somehow FLT3 ligand enhances selectively DC1 expansion, but we don't know the mechanism. Now, the last thing I want to talk about, just briefly, we've seen this slide several times about the controversy that we really don't know exactly what the cell biology of cross-presentation is. It's a very old and classic field, and we're all discussing how it works. One of the things about the cytosolic loading of class 1 is that it goes through TAP, the TAP transporter. Peptides are made by the proteasome, and they do go through TAP and are loaded onto class 1. Now, TAP, this is the life cycle of peptide transporter. So, peptides are exchanged, a peptide comes in, it goes to a cleft that lodges itself to transport, ATP binds, ATP uses this thing down here is called the nucleotide binding domain. It enzymatically changes the conformation and shoves the peptide through into the lumen of the ER, and then it recycles. So that's an ATP-dependent transport of peptides. The crystal structure of TAP is known in the last few years, and the enzymatic mechanism is also known. So there is a thing called the Walker-A motif, which contains a conserved enzymatic residue called lysine residue, which is critical for the ATP hydrolysis. So we wanted to ask the question, does cross-presentation require TAP transport? And so the way we did that is Luke, he made a new mouse. This time he mutated the critical lysine in the Walker motif in TAP2 to inactivate the functions TAP, because we didn't want to remove TAP completely. We wanted it to be present as part of the larger peptide loading complex with other chaperones that it forms. So he made this mouse, and sure enough, they don't cross-present. So TAP deficient, TAP inactive DCs don't cross-present. So what I'm showing you here, this is the standard OT1 proliferation assay that we all love. And if you see a lot of cells over here accumulating, that's the signal for cross-presentation in response to this heat kill Listeria ovo, which is a form of a cell-associated antigen that's DC1 specific. And over here, you don't see that signal, or it's greatly reduced, so that's no cross-presentation. So that says that's consistent with requiring peptide transport from the cytosol. But when you look at the DCs from that mutant mouse that don't have functional TAP, this is a level of class 1. Class 1 expression itself on the cell surface is highly TAP dependent. And in fact, it's so TAP dependent that these mice don't even develop CD8 T cells, which is required for their positive selection in the thymus. So there's no surface expression of class 1 on the surface of DCs anyway, so no wonder there's no cross-presentation. But we were aware of this old paper by Kitty Blue, where he showed the biology of MHC that is in a TAP-deficient cell, that the stability it relates to the stability of the empty class 1 molecule. And what he showed was that either keeping them on the bench overnight at room temperature, or adding peptides that combine with the class 1 molecule can bring out the TAP. And here's another paper a few years later by Ron Romain, in which he explored the mechanism by which peptides can stabilize the TAP. So we tested this. So the top two blue ones are just wild type, so that's the high level of surface expression on the normal DC. In the TAP2 mutant cell, they lose that here. But if we add a peptide, and this peptide sequence is here, VNF, that's called VL8 peptide. It's from SARS-CoV-2. We got it from Mike Diamond and Pritesh Desai. When we just add that peptide to the culture, then surface class 1 is restored. And it's all due to this peptide from SARS-CoV-2 in the group. Now this induction that I just showed you over here, this is very specific. It's specific because it restores K of B expression, but it doesn't restore D of B expression because it doesn't bind to D of B. So it's only restoring surface expression of cells, of molecules that it can bind in a group to. So what? Irrelevant exogenous peptides stabilizing K of B on the cell surface, they should be irrelevant to cross-presentation. No one said it. Well, let's see. So I showed you this. Now, when we add the peptide, the VLA peptide, to those dendritic cells that are TAP deficient, now we restore a signal. And it's, so is this cross-presentation or not? Well, it's not the peptide. It's not the VLA peptide stimulating OG1. It's the peptide from the cell-associated ovalbumin that is now being able to access the class 1 molecule somehow. And we did this with another version of TAP2 deficiency, here with a large dose response of the heat killed over. And you can see here's another a complete TAP2 mutant. The signal for cross B is pretty low, and if you add the VLA peptide, the signal is pretty large. And we did it a third way. There's a way to make class 1 MHC stable to being empty of peptide, and that's by making the 84 and 139 residues, turn them into cysteines so that they can form a double bond, a cysteine bond, and that stabilizes it so they don't fall apart. And that was published in this paper to show that it can restore surface expression of class 1 in a TAP knockout. And it does. So we here are using, in this case, it's a TAP1 deficient Lu2 cell, which loses class 1 expression, shown here. But expressing this CC mutant of KFB restores KFB surface expression pretty highly. And again, this kind of mutant, we expressed it in a TAP1 deficient Lu2 cell. Normally, here's the signal for cross B for Lu2, it's pretty good. TAP1 deficiency shown here, not so good. And when you re-express the mutated stable KFB into a TAP1 deficient Lu2, it really restores cross-presentation of the cell associated. So maybe that means that cross-presentation can occur in the absence of TAP, but we don't know. It's still a work in progress. The last thing I'll say is that this is a paper we published in 2018. We did a CRISPR screen for real cross-presentation in primary DCs with candidates, and we hit this gene called WEFY4. We have the knockout, and the knockouts absolutely don't cross-present in vivo. They can't get a tumor, they can't get tetramer to a virus, and it looks pretty real. This protein is greater than 3,100 amino acids. This is an alpha-fold imaginary structure, and it's huge. It's hard to work with, and what I've painted on here are the residues on the armadillo domain and other domains that are really highly conserved between lots of species. And so our imagination is that this protein has some kind of role in cross-presentation, but we don't have any clue as to what that mechanism is. We're working hard on it. We're not making very much progress, and it would be agnostic to whether its mechanism is cytosolic entry, lysosomal rupture, vesicular trafficking, or whatever. Now, Julie Blander is one of the other people in the field who has proposed that a vesicular pathway for cross-presentation exists. This would be a recycling compartment for class 1, and we didn't. This was, I'm stealing this from her commentary on RW paper. She had a diagram suggesting that WDFY4 plays some kind of role in the particular trafficking for a particular pathway. But anyway, the take-home message is that we have evidence that some DCs can cross-present even if they don't have CAT, as long as you restore class 1 in some artificial way. You still need to prove that that artificial way is a WB-dependent way. Because we think that WB is required for the real cross-presentation that happens in vivo by DC1. I should say that if you know how to do it, you can get a DC2 to cross-present, and that's like everyone, like everything Michael Douglas showed it first, which is if you form strong immune complexes with your antigen, DC2s cross-present very well, and it's WDFY4 dependent. So we need to work out the mechanism of WDFY4. So I think I'll just stop there, take questions, and thank the audience for being such a good sport.
Really amazing talk, and I suppose that there are going to be a lot of questions. I have one or maybe two. The one is, you are assuming that all these is transcriptional control. These are transcription factors binding to enhancers. Is it possible that the epigenetics also have a role by remodeling chromatin and specifying lineage?
Yeah. The question is, do you hear the question?
Oh, so yes, of course, yes. Yeah, they heard the question.
If the question is there a role for epigenetics in clinical modification? Yes, we think that's what happens. There are sites and factors that can be opened without help. And the word is pioneer factor, and that's kind of a loose, poorly defined term, but people use it. Then there's a whole talk that I skipped for time, which talks about how one enhancer influences the chromatin accessibility for the next enhancer. And I call that the F1 effect. I can show it if you want, but yeah, if you want.
Another polymorphisms in these enhancer regions, and does that affect the proportion of different dendritic cells generated in individuals?
That's a great question. One of the take-home messages from that first part with the high is that we don't know what the impact of enhancer alteration would be in polymorphic population. It could be pretty weird.
Do you know to what extent there have been comparisons of mouse strains?
I know that Michelle Newsensweig told me that his last project in the DC area was going to be a genetic analysis of different mouse strains, showing the different types of DCs that would be present in different tissues at different ratios and work that out. He told that to me a few years ago when I was talking to him. I've seen him a couple of times since then, and he hasn't finished the project, and I don't know if he'll ever publish it. But it's a very important problem. We ourselves found a big polymorphism. When we first were identifying a DC1 progenitor, our mice were on a 129 genetic background. And the DC1 progenitor doesn't stay at DC1 in vitro on that background, but it does in V6, so we never figured that out either.
Please.
Yeah, I have two questions. So, like in Martin's paper, the ZAP2 transgenic has also these middle cells, these like promiscuous cells. Like, how do you fit that into this transit expression model?
What, in whose paper?
Like in Martin's paper, the ZAP2 transgenic.
The hybrid DC. He's asking about the hybrid DC. A monster.
Like this middle population, which is XA1 and
I don't know that answer. I'm not sure I understood your question.
We can answer it. We see we're that simple.
Yeah.
More questions?
the second question is like, how do you put PU1 and the levels of PU1 into this model?
We almost ignore E1, I'm sorry to say. I mean, It's not because we don't think it's important, it's because we just can't do everything, and we haven't ourselves experimented with it. It's clear that P1 has broad impacts on both billboard and monitor communities. Ernestine is the one who did a lot of that work a long time ago, and I can't think of it.
So, Mike?
Thanks, Dan, it was great. Sorry about the croaky voice. It's good night. We know the course, yeah. As you might remember from our mini paper, the B Corp knockout does not have a phenotype in the steady state. And we noticed also from non-transplantation setting, there's no phenotype. As soon as you do flip-through ligand, you get funky stuff happening, right? So there's no difference. The other thing I know is that B Corp binds IRF8 in B cells. I don't know anything else, but could that do something in your
Did you say that the NFL3 knockout doesn't have a phenotype in steady state?
The
B Corp knockout?
Oh, B Corp, right. Okay, I thought you were talking
No, sir. B Corp.
Yeah, that could be the same kind of thing. The problem with trying to figure out a bypass pathway is that you don't have any place to start. I thought that it gets even more complicated. The CEPs, in our case, the CEP alpha and beta, these are odd genes because they're single exon genes that have translational control over either a fully active isoform or a short isoform that is negative. And since they heterodimerize, they can be squelching negative. They can snuff things out. So we thought that FLT3 signaling influences the metabolic state through mTOR, through ribosomal translation to favor the production of the negative form. So I had a postdoc make mice that have CEP long or short constitutively, and we crossed into the NFL3 knockout, and nothing happened. And that's because we used the wrong CEP. We did it with CEP beta, which have lip and lap. And so I'm trying to find someone to go back and make the CEP alpha equivalent isoforms, because I think that it's going to be FLT3 ligands favoring the short isoform of CEP alpha, firstly known studies, which will be a dominant negative form, suppressors that do just at the right time when you get DC2024. That's what I think. If anyone wants to come to the lab and do that project, I'll pay you.
So, more questions? I have one. Having read the Perforating 2 paper and seeing your data with the very elegant mutant, there could be peaceful coexistence of the cytosolic and the macular route of cross-presentation?
The issue of whether it should be peaceful, I think we just want to find out the way it works, and when it works, the way it works. So if something's working in some setting, that's good. The setting I think we would like to know is that when you immunize somebody with something, or they get a viral infection, and those DCs cross-present antigen, how does that antigen look? And I'm not biased one way or the other, but I think it's difficult. We all have the challenge of setting it up and testing it in vivo.
So thank you very much, Dr. Murphy, for a really amazing talk.